Okay, so welcome to this next video in which we are discussing the opening mechanism of the cis-loop ligand-gated ion channels. Okay, right, so we're in the process of discussing the structure of the cis-loop ligand-gated ion channels. We've now discussed the transmembrane region, we've discussed the intracellular domain. Now what we need to discuss is the ligand-binding domain, which is the extracellular domain of these ligand-gated ion channels. And basically, the ligand-binding domain is made up of this extracellular portion here, this pre-M1 region, the portion of the protein that's prior to the M1 region, i.e. the portion with this cis loop in it. Okay, right. So this is the portion that's actually going to form the binding domains for the ligand. Right, so let's now discuss the ligand binding domain. And the problem is that this varies largely between the different receptors. So what we're going to do is take two examples that are very, very different, and we're then going to see the structure of their ligand binding domains. Well, the actual ligand binding domains on them is pretty much the same, but the number of ligand binding domains is going to be different. And we'll then see how the opening mechanism in each of them works. Okay, right, so the two receptors that we're going to look at are both nicotinic acetylcholine receptor. So let's discuss more about the nicotinic acetylcholine receptor. So basically, we know the nicotinic acetylcholine receptor is a pentamer of these subunits that have this structure that we've been seeing all along. Now, the question is, how many different genes do you have which code for the subunits that can make up nicotinic acetylcholine receptors? Well, the answer is that you have 17 genes which code for protein subunits that can make up nicotinic acetylcholine receptors. So let me emphasize exactly what that means. To make a nicotinic acetylcholine receptor, you need 5 of these protein subunits. Okay, now which protein subunits can you actually use to make this? Well, we know they all have structures that look like this. However, what genes actually encode for the subunits that can come together to make a pentamer that is a nicotinic acetylcholine receptor? Well, basically there are 17 genes coding for proteins like this, which you can use to make the nicotinic acetylcholine receptor pentamer. Now, to help us get some sort of grasp of this number of genes, we have grouped them into families. So we have the alpha receptor subunits, okay, and there are 10 genes in this family. So there's the alpha 1, alpha 2, and then it goes all the way down to the alpha 10 gene, basically. So all of these genes will have slightly different sequences of organic bases, which will lead to them making proteins with slightly different sequences of amino acids, but they all have this same membrane-spanning topology when they're assembled into the membrane, and they can all come together in pentamers to make nicotinic acetylcholine receptors. Okay, the next family, that's 10 genes. The next family is the beta family, which has 4 genes. So this has the beta 1, beta 2, beta 3, and beta 4 genes. Okay, so again, these are all genes which will code for proteins which can make up a fifth of the nicotinic acetylcholine receptor. Finally, the 3 last genes aren't grouped into families, and they're just called the gamma, delta, and epsilon genes. Okay, so that is the 17 protein subunits that you can use to make a nicotinic acetylcholine receptor. So, the question is, how do you put these together? So, is it the case that you take for all 17 of these, that you can build nicotinic acetylcholine receptors by making 5 copies of each protein and sticking them together to make a homopentamer? Is that the only way you can do it? No is the answer. You can also make heteropentamers, and in fact some of these will not form homopentamers. So let's have a look at 2 types of nicotinic acetylcholine receptor, which are very physiologically important, because as you can see, this looks like a total nightmare. I mean, the number of potential combinations you can have is humongous, but there aren't that many that are actually notable as a student. If you're an expert, then you'll need to know about a few more, but as a student, there aren't actually that many that you need to know that are notable for their role in the human body. So we'll start with the most famous one, which is the one that's on skeletal muscle cells. Okay, right, so what's the subunit composition of the nicotinic acetylcholine receptor that's on skeletal muscle cells? Well, it's often referred to as the alpha-1,2 beta-1 delta epsilon nicotinic acetylcholine receptor. It would also be called the neuromuscular junction nicotinic acetylcholine receptor. So this tells us which subunits we're going to use. We're going to use two alpha-1 subunits, a beta-1 subunit, and a delta and an epsilon subunit. However, it doesn't tell us how to actually stick them together in a ring. So let me show you how they're actually stuck together in a ring. So here's one of these cartwheel drawings again, okay, with the pore in the middle and the 5 receptor subunits making up the pore like so. So the structure of the neuromuscular junction nicotinic acetylcholine receptor is that you put the two alpha-1 subunits like so, with the epsilon subunit in between them there, the delta subunit here, and the beta-1 subunit up there. Okay, so let's colour this in to make it look nice and interesting. So in purple, or outlined in purple at least, these are the alpha-1 subunits. Okay. Then in blue we'll have the epsilon subunit. Okay, so this is the epsilon subunit. Okay. The beta-1 subunit we'll have in orange here. And then in delta we'll have the green subunit. We'll have delta in green. Okay, so in green is the delta subunit. Right, so this is the neuromuscular junction form of the nicotinic acetylcholine receptor. Now the question is, where are the binding sites for acetylcholine? Because that's what we're studying now. We're studying the extracellular domain, and we want to know where are the binding sites. Well, this nicotinic acetylcholine receptor only has 2 binding sites for acetylcholine, which is in contrast to the one we'll see in a moment. Okay, and these 2 binding sites are in between 2 subunits, which we'll see as a common running theme. So there's one between this delta subunit and this alpha-1 subunit, and there's another one between this alpha-1 subunit and the epsilon subunit here. And I would also just like to note that this is the adult form of the neuromuscular junction nicotinic acetylcholine receptor. There is also a fetal form which has a slightly different subunit composition. So the fetal form of the neuromuscular junction acetylcholine receptor is the alpha-1,2 beta-1 delta gamma nicotinic acetylcholine receptor. So basically you just take this epsilon out and replace it with a gamma subunit, and then you have the fetal neuromuscular junction nicotinic acetylcholine receptor. Okay, right. So you have these 2 acetylcholine binding sites. So these are acetylcholine, ACH for short, binding sites. Okay. And we'll study the structure of this in a bit more detail in a moment. But firstly, I just want to show you how this can be totally different, because if we look at the alpha-7,5 homopentamer, so this is a heteropentamer because it's got loads of different types of subunits all put together. So this is a heteropentamer, whereas the next form of the nicotinic acetylcholine receptor that we're going to look at is what's known as the alpha-7,5 homopentamer, okay, because it's made up of 5 identical subunits, all of this alpha-7 form. So basically, if we now draw the structure of this, it's very simple, because in each slot you now just have an alpha-7 subunit. Okay, so here's the receptor, here's the pore. Let's divide it into these 5 portions, and then you just have alpha 7 in all 5. So here's alpha 7, here's another alpha 7, yet another alpha 7, and then finally 2 more alpha 7s. Right, so the acetylcholine binding site for these alpha 7-5 nicotinic acetylcholine receptors is again in between 2 subunits, and from that you should be able to guess how many of these acetylcholine binding sites you're going to have just by the rotational symmetry of this alpha 7-5 homopentamer. It's got to have 5 acetylcholine binding sites. So 1, 2, 3, 4, 5. 1 in between every single alpha 7 subunit. So 1 at every single intersection between 2 alpha 7 subunits. Okay, so this alpha 7-5 homopentamer of nicotinic acetylcholine receptor is not found in skeletal muscle cells. Instead, it's found in the brain. It's one of the 2 very common nicotinic acetylcholine receptors that you have in the brain. Okay, it's also very involved in the addiction process to nicotine. So nicotine can also bind to these acetylcholine binding sites on the alpha 7-5 homopentamer and cause it to open, and basically this receptor is on the ventral tegmental area within the midbrain, and that causes activation of these ventral tegmental area neurons, and they then release dopamine into the nucleus accumbens, leading to positive reinforcement of the behavior which gave rise to the nicotine rush in the blood, which was the smoking. Okay, but nicotine does not bind to the acetylcholine binding sites on these skeletal muscle nicotinic acetylcholine receptors. If it did, it would cause involuntary muscle contractions, which could actually be fatal. So people wouldn't smoke if it did. So it's not an agonist at the skeletal muscle form. Okay, right. So we've now seen the fact that you can have a different number of acetylcholine binding sites, and this is going to be important when we discuss the opening mechanism. Okay, right. So now let's have a little discussion of the structure of these binding sites, and then we'll look at how when ligand binds it causes the acetylcholine receptor to open. But we'll continue this discussion in the next video.